Okay, we're going to get started. I'm Shannon McQueenie. I'm the division head for bioinformatics and computational biology in DMICE. Dr. Hirsch, who normally does the introductions, is away on travel, so I have the honor today. We're here for a proposal defense in which the students are going to be presenting the ideas for their thesis. Brandon Keibler, who's to my left, is one of our master's students in bioinformatics and computational biology. He had done his bachelor's degree in bioinformatics at Pacific University. He's a second-year student, and he'll be talking today about work on semantic workflows for neuroimaging. He's also involved in another collaboration besides his thesis, which is on transposable elements. If you're online at home or wherever and want to tweet questions, we'll be monitoring Twitter, and the key hashtag is DMICEConf. It's DMICECONF. All yours.
Welcome, everybody. As you said, I'm Brandon Keibler, and this is my proposal for semantic workflows for neuroimage processing. So neuroimaging is, of course, taking images of the brain, in our case, usually a human brain, and just something to keep in mind. Often when we're looking at these images, we are seeing two-dimensional representations of the brain, as you see in the background of this image. Just keep in mind that that's usually just one single slice, one single view of a much larger three-dimensional model. And if I do ever use the term voxel in this presentation, just know that that's essentially a three-dimensional equivalent of a pixel. So how do we get these images? Mainly this is done through magnetic resonance imaging, because this allows us to get very high-quality images of the brain in a very minimally invasive way. Basically, the way that this works is the subject is placed inside of an MRI machine, or in our case, perhaps just their head, and then this large magnet creates a strong magnetic field around the subject. Basically, this is going to leverage the fact that our bodies are made mostly up of water. So this magnetic field that's created causes the protons in the hydrogen atoms of the water molecules to all line up and orient themselves in the same direction. From there, this radio frequency coil emits a radio frequency that excites the protons, and then it's turned off, and as those protons return to equilibrium, they release their own radio signal, which is then picked up by the sensor. And this is most commonly used clinically to detect irregularities in a subject's brain. You can see here in this example it's being used to identify the presence of a brain tumor. You can see how that brain tumor expands over the course of time. And this is done just using structural images done by a standard MRI. It can also be used to incorporate functional MRI, called just fMRI data to show activity in the brain. In this example here, you're looking at a child who's currently undergoing an epileptic seizure, whereas the warm colors indicate portions of the brain that are more active as compared to the resting state, whereas the cooler colors indicate areas of the brain that are less active. And so this is basically done by using something called BOLD imaging, which stands for blood oxygen level dependent. And the basic concept here is that as a part of the brain becomes more active, the body responds by increasing the blood flow to that part of the brain, which correlates then to higher concentrations of oxygenated blood in that area. And basically there's some differences in the magnetic susceptibility of oxygenated and deoxygenated blood. And the MRI machine is able to pick up on that. So essentially the way an image like this, in the seizure example, how that image is created is they first take a structural image of the brain, which usually takes an amount of time, somewhere around an hour, to create that high resolution image. And then the functional MRI pictures, which are only interested in the brain at particular points in time, are taken. So these are much faster snapshots taken that result in a much lower resolution image. Well, then basically you take the functional MRI image at one point at the resting state and compare it to the functional MRI image while the individual is undergoing, in this case, the seizure, and they're compared to identify what part of the brains are more active or less active, and then they're color-coded as they are in this example. And then those functional images are then superimposed on top of the structural image of the brain so that we can actually see where that differing levels of activity are taking place. Another way this can be used is in connectivity mapping. So in this case, we are looking at how the activity of different parts of the brain correlate with each other. So it's essentially done by taking a time series of these functional MRI images, and you look at one particular point of the image and see how that signal changes over time, which results in this graph mapping out the brain activity pattern. You then look at another part of the brain, a similar graph, and then compare them to see how well they correlate. So in this example here, we're looking at individuals with and without ADHD, and this is looking how all of these different parts of the brain, how they correlate with another part of the brain called the nucleus accumbens, which is right about here between the orange and the blue. And so areas of the warm color, that's where when the activity of the nucleus accumbens increases, they also increase. And their activity patterns are somewhat correlated with each other. The cooler colors indicate areas where the activity of those two brain regions are inversely correlated. And so probably the most highest profile example of this kind of work is the Human Connectome Project. It's a large collaborative effort to map the human brain. And so it basically has established a foundation of the functional anatomy of the brain and what variation in that exists in the human population. One of the things to come out of this is the Human Connectome Project minimal processing pipelines. So this is essentially what they've established is a standardized method for taking the raw MRI images and processing them into prepared data that's ready for analysis. And there's a lot in here that I'm not going to get into. The general idea is they take their structural images and prepare them, which in and of itself is composed of 3 separate pipelines, working one after the other. And then from there, they can incorporate their functional data, which in itself is processed, or they can bring in, they also have a pipeline to bring in diffusion-weighted images, which is a whole other thing that I'm not going to get into. Basically, we're going to just be looking at the very first step, which is called the free surfer pipeline, named thus simply because it comprises all of the processing steps that take place before the next pipeline, which uses the free surfer software package. This is a very, very simplified map of what's going on here. There's a lot of steps that I'm not showing. But basically, this pipeline is focused on 2 separate tasks. One is correcting the images for various levels of distortion, various sources of distortion, as well as a bias field, which I'll talk about more in a bit. That's basically a signal corruption that comes up with MRI imaging. And then the second goal is to align the individual's brain to a reference brain template. So we start off with our brain images. Here we're showing that there's basically 2 versions of this, a T1 weighted and a T2 weighted image, which are 2 separate images that are taken ideally around the same time, that are just taken using different settings on the MRI machine. You can kind of think of these as taking a picture with or without the flash on. And the T1 weighted image is kind of what is more focused on. The T2 weighted image comes into play because we found that it basically helps inform some of the processing algorithms. Basically, the different levels of contrast between the different parts of the brain in these two images, that information can be used to help get some more accurate results from different processing steps, such as separating out the white matter and the gray matter from the brain. The T2 weighted image, like I said, the importance of this has kind of been recognized a little bit more recently. There's certain situations where that might not be available, such as if you're looking at data from an older study, might not be present, but ideally you'd have both of these. And then what you're getting out of it is basically two different images. The original brain on the far left there, which is corrected for processed brain image for a particular individual. And then a separate version of that, which is aligned to a reference brain template in a process called registration, which I'll go into more detail in a moment. But basically, the original individual's brain, everyone's brain has unique size and shape to it. And so that's reflected in that brain image. And then the registered brain is basically the same brain made to conform to the size and shape of the reference template. And that would be the appropriate version to use when doing cross-subject comparisons and such. So depending on what you're doing downstream, you would use one of these two images.
I have a question. Can you go back a slide? Yeah. So you said that the reference brain conforms to different shapes and structures of the original brain, the study brain?
No.
Can you say that one more time?
Sorry. The original brain. This is the individual's brain in what's called their native space. This is their natural shape of their brain. And then the reference is basically just a standardized brain shape that the original brain is then transformed to conform to that same shape, so that if you're looking at two separate individuals, this part of the brain is going to be in the same place for both of those individuals. Okay, so the first kind of distortion that this pipeline corrects for is called gradient distortion. These are distortions that arise due to imperfections in the magnetic field gradient. Basically, the yellow crosshairs here is the isosender where the scan's being taken. And then as you can see, the further away you get from that, the stronger, I don't know if you can see the red lines, basically the further away you get is the stronger the effect of these gradient distortions are. And you can see in this example, before the correction is done, the original image seen most obviously here in the neck, where it's kind of squished together, whereas after you do the correction, you end up with a neck that's more accurate to the actual anatomy of the individual. So this is a step that's recommended but somewhat considered optional. It kind of depends on the MRI machine that's used to acquire the images. Some of them are more susceptible to this type of distortion than others. So in a lot of cases, this can actually be skipped. Another type of distortion, which is comparatively minor, is called readout distortion. And this basically arises from varying magnetic susceptibility of different tissues. And so this is corrected for by using what's called a field map, which is something that's acquired ideally immediately before acquiring the structural images of the brain. And there's different ways you can create a reference map. But basically, it's going to be a file that it's a lot better. It's going to be a file that basically just defines where this readout distortion is occurring in the image. As you can see here, this is an example where two images are taken using different readout spin directions. A little more technical than I want to go into, but basically these two images are taken before actually acquiring the structural image, and you can see that there's slight differences, probably most noticeable right here, where the picture's a little bit different. So basically, these two would be used together, and then where these differences occur is going to inform in the field map where this readout distortion is occurring, and then that can then be used to correct for that in our structural images. But yeah, like I said, there are a couple of different ways you can create a field map, which again might not always, under certain circumstances, might not be available. And if that's the case, you'll likely have to skip this distortion correction, which can be okay depending on the sensitivity of the downstream analysis you're doing. But it's again something that you're going to want to do. A bias field is not a distortion, but it's basically a gradual signal gain in the image that's caused by the variations in the magnetic field, as opposed to the actual anatomy that we're interested in. And this is where that T1 and T2 weighted images come into play. Essentially, you can combine the T1 and T2 weighted images, and effectively they cancel each other out, and then what signal is left over is going to be resulting from that bias field. And you can take that then, subtract that from the structural images to remove the bias field corruption. And like I mentioned a bit earlier, the T2, under certain circumstances, that T2 weighted image is not always available, in which case there are alternate methods to correct for this to estimate that bias field that don't rely on the T2 weighted image. That can be used in those cases. And then the final step is registration, which I mentioned a bit already. Basically, it's a nonlinear transformation to transform the individual's brain to conform with the reference brain template and get you that standardized brain shape. So, and this is another place where the reference map that you're using, that's going to be informed by what kind of study you're doing, what kind of subjects you're working with. Normally, we would just use the standard adult human brain reference. If you're working with different animals, you're going to want to use a reference that's appropriate for that species, for example. But basically, this pipeline is provided as a shell script, or a group of shell scripts that are designed to be run from the command line. And it generally comes out looking something like this. But obviously, there are a lot of things that you can do to make this a lot more manageable, but if you were to run just the pre-free surfer pipeline, which again is just the very first step in this process, and run it straight from the command line, you'd be typing in something along these lines. And there are even more parameters here that I'm not showing, and the situation, exactly what you're doing, and how you got your data kind of determines what parameters you have to use, and some of these, under other circumstances, wouldn't even come up. it wouldn't be included here. But basically, what I want to show is that this is kind of a mess. It's hard to kind of keep track of what's going on. It's certainly vulnerable to typos and such. So this is kind of becomes a concern as far as reproducibility. Reproducibility being kind of a hot topic in science at the moment. Essentially, some studies have been done to show that when they try to reproduce in high-impact studies, they end up getting different results. These estimates vary, but some estimates put the amount of preclinical research that's simply not reproducible being somewhere around 50%. Cancer being especially prone to this, with 89% of it being irreproducible. And that kind of begs the question, if we are not able to reproduce our studies, then how much can we really rely on their findings? So this kind of translates into a lot of time and a lot of money being spent on studies that we can't really trust. And so one way this has tried, people have tried to address this as far as running pipelines and data processing goes, is to use workflow management systems, which essentially attempt to make this process a lot more accessible and a lot more user-friendly by doing things such as providing a GUI interface and providing visual representations of the workflow. And so one example, which is used pretty heavily in the field of genomics, is Galaxy. Basically, it's a free web-based tool that just about anybody can access and have access to a large library of tools commonly used in genomics without having to install any of the software on their own system. And then, of course, this allows you to arrange these different tools, select them, and pipe them together into a coherent workflow to process your data. Another example that's much more focused on neuroimaging is called Lonnie Pipeline. It's some standalone software, but again, this comes with a pre-created library of processing tools that are used commonly in neuroimaging. Allows you again to put together your own workflows and even visualize as your data is being processed through the various steps. And so this is just a couple of them. There are tons and tons of workflow management systems out there, and they all have their own benefits, but they all have their own limitations as well. So using these workflows effectively still requires a certain level of knowledge about the workflow from the user, specifically regarding what algorithms are being used, the different parameters that are involved in these algorithms, and what would be appropriate values for those, as well as any assumptions that they have about the data that you're inputting to be processed, which can go a lot farther than just whether it's the right file type. Additionally, if you're putting in multiple input files, then there's often assumptions about how those two inputs are related and any interdependencies that they might have between each other, which often aren't explicitly shown to the user. So one thing that's kind of come out to try to address some of these limitations are semantic workflows. And basically, why they stand out from other workflows is that they encode a certain level of knowledge about a workflow into the workflow itself, which it does by implementing what are called semantic constraints. Basically, semantic constraints are there to provide an extra layer of error checking and rule enforcement to help a user operate a workflow as it was intended. So this is kind of some examples of what these semantic constraints look like. In our precursor for pipeline, the very first step is the gradient distortion correction, which again is in certain cases optional, but is highly recommended for certain distortion. So information, some metadata about the files, including who this brain belongs to, when the image was acquired, and what machine was used to acquire it. And it can pick out whether or not that gradient distortion correction is especially necessary. And in this case, recommend that it be used where it might otherwise just be skipped. Another example in genomics, when you're doing patient variant analysis, you basically got a file showing the DNA sequences for an individual, and you want to compare that to a database of known DNA variants to see which of those variants your patient has. Both of these files were created by aligning DNA reads to a genome assembly, a reference genome, reference human genome, of which there are several versions. And if those two files were made using different genomic assemblies, then suddenly the genomic locations referenced in this file aren't going to correlate with the genomic locations referenced in this file, and the whole thing's going to fall apart. So in this case, we'd want our semantic constraints to identify this issue and prevent this workflow from being run. Input compatibility, like we saw with the previous example, it allows to restrict selectable input. So once a user selects one input file, when they go to select the next one, it's only going to show available inputs that are compatible with the priorly selected one. It can also recommend inputs and parameters to the user based off information it's getting from the dynamically assigned metadata to your files, so that your output files automatically have metadata that describes what kind of processing steps were used. It can also be used for some automatic processing path selection. You've got two different algorithms that are used for the same process that are used in different situations. If there's any way that you can encode that those situations into the input files, then semantic constraints can be used to automatically recommend or select which algorithm to use. So a workflow management system that allows the use of these semantic constraints, and the one that I'll be using in my project is called WINGS, which stands for Workflow Instance Generation and Selection. It's somewhat similar to Galaxy in that it's a web-based tool that you can access from anywhere. Basically, you can select your workflows that you want to run, pick out what input files you want to put through it, see a representation of how the data is going to be processed and run it. One other thing this allows us to do is you can basically define these steps, or what are called modules in WINGS, to carry out certain, an individual algorithm, processing algorithm, or even a series of algorithms all in themselves, basically providing a certain layer of abstraction so that the user doesn't need to necessarily concern themselves with all of the nitty-gritty details and just focus on the general steps being used. So given this, I hypothesized that the operation of a semantic workflow for medical image processing will enhance its usability, and I plan to test that using these aims. First, the development of a semantic workflow to process brain MRI images, and then to evaluate the usability of said workflow. So the first step is obviously to make it. I'll be implementing a semantic version of the pre-free surfer pipeline in Wings, and while doing so, I'll be being sure to implement semantic constraints to protect from possible sources of user error, and kind of guide the user through the handling of the processing pipeline. And ideally, through this, we can enhance reproducibility as well. So some of the semantic constraints that I plan on implementing. First, whether or not the gradient distortion correction is necessary. This can be determined by looking at what type of MRI machine was used during data acquisition, and make recommendations as to whether or not that step's necessary at all. The use of the readout distortion correction. In order to do this, obviously we're going to need the field map that defines the readout distortion. So if that's not available, that's one step that we would need to skip. Alternative bias field correction handling. Like I said, we often don't have that T2 weighted image available, in which case we can recommend different processing algorithms to handle this step, or we can, if necessary, skip it altogether. And then also the compatibility of the reference brain map and the subject data. Obviously, if we're looking at non-human primate, we're going to want to use a reference brain map that's appropriate for that individual. And then the second step is to evaluate the usability of our semantic workflow by doing some user testing. So we plan to recruit 5 experienced users and have them use our workflow using 4 different sample data sets that we've put together to kind of highlight the different abilities, different benefits of semantics. And then once they've done all that, they'll report on their experience using a short survey afterwards. So the 4 data sets that we plan on having people use, basically the first one is just a normal, very straightforward data set that will allow users to kind of get accustomed to how you can use Wings. It can kind of illustrate how the different parameters can be restricted or recommended. Second one is missing data. Again, an example would be if we don't have a T2 weighted image, the workflow can then recommend an alternate method to do the bias field correction. The third data set would just be alternate handling, looking at potentially a young, data set, a brain image of a young child or a non-human primate, in which case the workflow can recommend an appropriate reference template to use. The last one being mislabel inputs. So in a situation where if you have a T1 and T2 weighted image, and through some previous error, one of the two is mislabeled as belonging to an individual other than the one it actually belongs to. We can use semantic constraints to try to look at the metadata of those files, whether it's the date that the images were taken or the age of the subject, and try to catch these inconsistencies and then provide a warning to the user to allow them to detect the problem. And then, so like I said, when they're done, we'll take a quick usability survey that will be based on an established survey called the System Usability Scale, which is comprised of 10, actually not questions, but 10 statements that the user will then indicate whether they agree or disagree with it. And then the responses from those can be used to calculate this overall score, which can give you a kind of a general idea of how usable the system was. And then in addition to that, we'll also be including some free response questions to allow for more specific feedback regarding their impressions of the semantic constraints, as well as bringing up any concerns they might have about the workflow that I might not have accounted for. So I'd like to thank my thesis advisory committee, as well as the members of the FAIR Neuroimaging Lab, particularly Eric Earl, for taking the time to answer all my questions, as well as the DMI staff for facilitating everything, and also my fellow graduate students who have helped brainstorm and talk me through this process. Any questions? Yes.
So I assume when you get these images, you get like a big stack of them from trying like walking through different slices of the brain. Is there anything between the different stacked images that you would want to keep track of and learn from in parameters for running it?
Can you repeat, I know this is a lot, but can you simply summarize or repeat so it's captured, because I don't know that they'll hear him.
Yeah, he's asking, so since these images are, brain images are three-dimensional images, saying if they're essentially a stack of two-dimensional images correlating to all the different slices of the brain, he's asking if there's anything in between those slices that we want to keep track of.
You want me to help with that one?
Yeah, the quick answer is not that I'm immediately aware of.
Yeah, there's in the majority of the way data is collected, there's not a whole lot of things that you care about in between the slices. But with that said, some of the new data acquisition procedures that make things really fast, what you do is you can take, instead of capturing like one slice at a time that go into the brain, you can capture what's called multiband imaging. You capture lots of slices all at the same time. When you do that, you can get this what's called slice leakage, where you can get some information from one picture that's taken, just like 20 milliseconds to the next one, just like the next 20 milliseconds. That folks are looking into to determine what that needs to be tracked or not. That's not something that's part of this yet. And likely for anything that we would be doing here, it's not going to be a problem because we don't collect that many slices at a time. But it's a good question.
So like the normalization parameters that you use from slice to slice, change, or do you as you're adjusting each image, you do that all separately?
Well, you look at all of them together. Okay. What he was talking about with some of the bias corrections, stuff like that. You're looking at all of them together to determine whether that, how much that exists, how much you have to correct, but not like one at a time per se. You're using all the information.
Yeah, so that's what I'm wondering about in the workflow. Like, are you feeding it one image at a time, or you give it the whole stack and you learn some things from that, and then it goes back for each image?
Yeah, you're giving it the entire three-dimensional model as just one file, one three-dimensional image. I'm just looking at it all at the same time. Yes, please.
On the same topic, there actually is an error, at least on CMU scanners. The user sends the data to the server incorrectly, despite many, many reminders. They can do one of two errors. One of the slices, if they zoom in and zoom out to look at it and see it, if they send it wrong, that slice will be zoomed in, and it will be copied on the server. So instead of 160 slices, 161. And so a lot of programs don't recognize you have an extra piece of information. It also happened that the name of that file for that particular slice is the same, and they can actually replace it. In that case, you actually need to go in and zoom in or out and change it. And so that's huge, huge headache.
So, but there's no, is there any instrumentation area to indicate if that's happened? You're saying, like, so in the end I end up with an extra image, but what could I look at in terms of metadata or an error or something that would let me know that that's happened?
In the case of the copied image, you can look at the metadata you're talking about. The name of the actual diagram.
The name, got it. The name would give it away.
The good news is that won't happen anymore because it's been automated and doesn't work like this, so like there's no it's not, it won't happen anymore. The new scanner.
Yeah, but it's out there and interesting.
So when you're producing your aligned image by combining your reference with your original, can that process be reversed? So I'm kind of asking if it's a lossy conversion or a lossless conversion.
Well, you have the original image, so you wouldn't necessarily, ideally, need to convert back because the original image is saved. But then it also produces files that describe what transformations were taken to go from the original brain to the registered one. So I would imagine that could be used to...
The reason I'm asking that question is because if you're looking at two original images from two different people, and then you're mapping it to this combined image, I'm wondering if, like going back and forth, that this region in one image A is equivalent to this image in image B. So if it's lossless, it would be. If it's lossy, it might not be.
You're talking about for comparing images?
Yeah.
Because that's going to be a totally different issue.
I was just curious about it.
If you're going backwards, there will be some information loss because by necessity, we think about how it's a good example of this. When you're switching the reference frame, by necessity, you have to kind of interpolate the information to the new reference frame. And there's ways that we do that, and that's been tested enough anyways. So if you were to try to take that image and kind of make the inverse matrix and put it back into the original space, it wouldn't be the identical image. Okay? So I think that's what you were asking. It wouldn't be the identical image because you've changed, you've interpolated the data.
Yeah.
But like it says that when you still have the original image. So I don't know. It's
And
an
then
interesting question. But
there would be a file that describes, the transformation, so you can then use that to correct, one point in the registered brain and look at where that would correlate to in the original.
Any other questions? All right, so let's jump in. Thank you very much.